Hello and welcome to Module 1 of the course on Exploratory Data Analysis. This is Session 1 of Module 1. The session is titled Exploring Dependent and Independent Variables and Their Distributions. In this session, we will be talking about the concepts of independent and dependent variables. We will explore examples of these variables in health research. We will explore the levels of measurement of these variables. And we will try to understand how our population is distributed with respect to these variables. During this session, you may often have a feeling of déjà vu. For those of you who have formulated their research questions and did some literature search in order to assess the current level of evidence with respect to their area of interest, may have often come across terms like independent, dependent variables, response, outcomes, research design, etc. Now, it is but natural because most of health and well-being research focuses on studying a health or well-being outcome or multiple outcomes in a specific population of interest, and how certain factors such as environmental, biological, or social determinants of health are associated with the outcome of interest. For example, look at this study, which talks about the effect of nutritional promotion intervention on dietary adherence among type 2 diabetic patients in a specific setting. So, the target population of interest are type 2 diabetes patients. The factor they are trying to study is nutritional promotion intervention, and they are trying to look at how this factor affects dietary adherence, which is our outcome of interest. The way they would like to achieve an understanding of this effect is through a research design, the quasi-experimental study design. If you were to read this paper further, the method section talks about the measurement of the outcome variable, which the title says is dietary adherence. They also talk about in this section about measuring dietary adherence through a questionnaire. In addition, they also talk about measuring the fasting blood glucose level of the patients. So they are looking at multiple outcomes. Maybe fasting blood sugar is a proxy measure for assessing the outcome of interest, which is dietary adherence. We can understand here itself that the outcome is measured in two separate ways, and the way it has been measured in terms of the nature of the outcome data is also likely to be different. We can see the results section of this paper, which talks about the percentage of dietary adherence in several categories, and also how the fasting blood sugar is a measure of uh, the effect of the intervention, which is the nutritional promotion. So, in statistical parlance, the outcome or response is called the dependent variable. And the factors we are trying to study are called the independent variable. Dependent variables are the outcomes or effect that are measured in response to change in the independent variable. These are the variables that researchers are interested in understanding or predicting. What are the independent variables? The independent variables are factors that are manipulated or changed by the researcher so that they can assess the corresponding change in the dependent variable or the outcome. Let's take this example. Let's say we have three pots, each with a single leaf in the plant. The researcher decides to provide uh, water to these plants, but the amount of water given is different. The first pot of single leaf receives some amount of water. The second pot receives more than the first pot, and the third pot with the single leaf receives much lesser than the first pot. After a couple of days, the outcome of interest, which is the number of new leaves that sprout, is observed. It is seen that the first pot has many more leaves than the single leaf. The second pot does not have a single leaf. The one leaf has also died. And the third pot has an additional new leaf. So all the three pots started out the same, but the intervention that they got, or the independent variable over here, which is the amount of water that they received, was manipulated by the researcher so that they could see, given the same baseline and different forms of the intervention, how does the outcome vary, and can there be a pattern in this relationship. So the independent variables are input or causes that are hypothesized to affect the outcome of interest or the dependent variable. So here the hypothesis perhaps was that giving larger amounts of water probably destroys the plant, or a more exploratory hypothesis which says, how does the number of leaves change if you vary the amount of water that is given to the plant? Some other examples of outcome or dependent variables in health research are the blood pressure, weight loss or gain, pain levels, cognitive function, and disease progression. Independent variables, as compared to dependent variables, can be manipulated by the researcher. Dependent variables are measured or observed. The change in dependent variable corresponding to the change in independent variable may be causative or associative. The way we can determine whether it is causative or associative could be either by choosing an appropriate research design, or at the analysis stage by using an appropriate statistical analysis method. The research design can also determine whether we would like to study the association between the independent and the dependent variable as a causation or a mere association. Experimental design or experimental studies allow you to manipulate the independent variable to observe the effects on the dependent variable. So for example, in this study, which is a trial on the effect of a therapeutic gardening on mental health and well-being, By varying the levels of exposure to therapeutic gardening, the researcher can thus manipulate the independent variable, which is this intervention of therapeutic gardening, and observe how mental health and well-being outcomes change. In observational studies, on the other hand, researchers may observe um, the um, the naturally occurring relationship between variables without intervening. So, in this study, which is an observational study in a hypertension treatment and patient outcomes in a primary care setting, the paper says that they looked at data collected in the standard care setting retrospectively and looked at outcomes of blood pressure control. So the dependent variable over here would be, let's say, the percentage of participants having perhaps less than 140 by 90 blood pressure, um, and um, the kind of hypertension treatments that they received, which would be observed based on the records. Now, in order to understand in depth the relationship between the independent and dependent variables, and to choose analytical techniques that will help us determine the level of association or causation, or allow us to study causation, um, uh, uh, are two domains that help us understand the relationship better. These domains related to the data are the data types and level or scale of measurement and distribution of the data. So, so the outcome of interest is or the dependent variable is something that we need to investigate further and understand much better in terms of the scale of measurement of that outcome variable, and how the participants that you're studying are distributed with respect to that dependent or outcome variable. That is what is called data DNA, and that a better understanding of the nature of these variables allows us to use the appropriate analysis techniques. Now, there are two major types of data. Qualitative data, which is more descriptive in nature. These could include variables like color, uh, color of a mass that you are studying. For example, the color of the eye in a certain disease condition, the taste of some food that is understood to be nutritional, whether it is bitter, sweet. So the variable taste is described in terms of these categories or descriptions. Similarly, disease status, whether the patient is alive or dead after receiving a certain treatment, whether a treatment was successful or whether it failed. Um, so these kind of data are called qualitative data. Quantitative data, on the other hand, are numeric in nature. They may be finite numeric numbers or discrete, such as numbers like 2, 4, 6, where there is no other possible value of the variable between 2 and 4, and more continuous numeric variables um, uh, uh, such as a variable taking value, any value in the range 2 to 4, and so on and so forth. Let us talk about scales of measurement. Suppose we are looking at an outcome such as presence or absence of a disease. For each participant in the study, we would record the observation as present or absent. We may perhaps also assign a number 1 to present and number 0 to absent, or the number 1 to present and the number 2 to absent. We may have an outcome such as yes or no, where we record a yes as 1 and a no as 0. Such data where the outcome has a description and the assignment of numbers is merely for computational efficiency is called a nominal scale of measurement for the data. We may have an outcome such as in an opinion poll where a question is asked about the performance of a candidate, and the responses could be on a scale of 1 to 5, where 1 may represent strongly disagree and 5 may represent strongly agree. Now, the categories suggest some kind of an ordering from a lower to a higher order. Another example is the musical scale, where 1 may represent the lowest octave and let's say 8 may represent the highest note or octave. Such kind of a scale of measurement for data is called an ordinal scale. Suppose we are measuring temperature. Temperature is called an interval scale. Most common variables in biological research, for example, height, weight, and other such biological measurements like blood pressure, blood sugar levels, insulin levels, belong to the ratio scale. Other examples of variables that are quantitative in nature are height, weight, blood pressure, biochemical measures, scores on a test. We could also have numerical data such as the number of neonatal deaths or in facilities in the past 10 years. Here, the outcome would be discrete numbers such as, let's say, 4, 10, 6, 100, and so on and so forth. These numbers are discrete in nature. Outcomes like treatment failure or success, behavior adopted, yes or no, adherence based on certain cutoffs that are continuous in nature, um, approval or agreement scores on a, um, on a discrete scale of let's say 1 which is least and 10 which is the most level of agreement are all numerical scales of different types. Now, the second important component in understanding data so that we can apply the appropriate statistical technique is the distribution of the variable. A distribution of a variable informs us about how frequently, relative to the whole, unique values or range of values of the variable occur in the data set. So for example, for categorical or qualitative variables, variables that have categories, such as in this opinion poll, um, you can represent this variable graphically to understand its distribution using a bar graph. Here, each category has a bar as high as the number of people that responded to that particular category. So, for example, the maximum number of participants in your study could have opted for the strongly agree option in this particular question of the opinion poll. A good way to describe or summarize this data would be to use something called a modal value, which is the most frequently occurring value of this variable in your data set. In terms of discrete numerical variables, again, the bar diagram is a good way to summarize the distribution of the variable. Here, the outcome or dependent variable may have numerical values, but they are discrete in nature. So here again the frequency distribution in this example, which is the number of days people took their blood pressure measurements, can be represented by a bar diagram. Here uh, the graph shows that approximately 8 people took blood pressure measurements on 4 days. Approximately 15 people took blood pressure measurements on 36 days. This distribution can be characterized by an average value, which will tell us the average number of days that people took home blood pressure measurements. It could also tell you the range of values, which is the minimum and the maximum value, and the spread of this distribution in terms of the standard deviation, which are parameters that characterize this distribution. For continuous numerical variables, say for example, blood pressure as the dependent variable or the outcome variable of interest, we would have represented them as a histogram, which is a frequency distribution of a continuous variable. In such a diagram, the x-axis is consisting of the values of interest, and the y-axis tells us about the number of people who had that specific value. However, for continuous outcomes, the range of unique values may be very wide because there are an infinite number of numbers that exist between any two numbers in a continuous variable range. So, again, the height of each bar represents the number of people who had that particular value of the continuous variable. However, in this case, because of the infinite number of possibilities between any two values of a continuous variable, it makes more sense to understand this distribution in terms of the number of people who had values in a specific range, below a specific value, or above a specific value. Such kind of a distribution is also called a frequency curve when you overlay a smooth curve over the distribution, just as is shown in the right hand side of the figure. This gives a sense of the shape of the distribution, and the area under the curve can also be interpreted as the probability that if a person chosen at random from this population would have a value of that variable in a certain range below a specific value or greater than a specific value. So, this kind of a frequency distribution can be interpreted not only in terms of the number of people, but also the probability of an event where the event is described as a participant having an outcome value in a specific range. There are several kinds of distribution that exist in medical science. The one that we saw previously seems to be more symmetric, where the left-hand side of the distribution is very similar to the right-hand side if we were to draw an imaginary line at the center. Also, the previous distribution can be characterized by parameters like the mean and the standard deviation, which represents the central or the average value and the spread of the distribution. On the left hand side of the screen, you see different kind of distributions that are possible. You will see distributions that are asymmetric. Some distributions may tend to go towards the left-hand side of the scale. Some tend to be more stretched towards the right-hand side of the X scale. Such distributions are called negative and positive skew, respectively. Similarly, there are different shapes for discrete distributions also. We will hear more about exploratory data analysis and the distribution of variables in the live session. Thank you.